So I will be talking about 3D Strudel, which is a tool for local map model validation in cryo-EM. So I will try to keep it simple, so you don't really need to be an expert in cryo-EM to follow it. So why were we working on that? That's cryo-EM, as you all know, it's growing fast. And with these numbers, it's really important to have good validation tools to keep the data of high quality. So to tackle this, over the years, there were organized several validation task forces where basic principles and the methods for validations were formulated. And at one of these meetings, it was stated that visible features should be in accordance with the claimed resolution. But what does this mean? So, for example, how should an amino-acid residue look like in cryo-EM? Here I have 6 images of phenylalanine from several maps, which are all at 2.5 angstrom resolution. So how should look like really a phenylalanine? Because as you can see, they are quite different. And in reality, none of this image is the true image because this method is affected by a high level of noise. But things can be improved quite a bit by doing simple averaging. And for this image here, I have averaged 80 images of phenylalanine and 2.5 angstrom resolution. And as you can see, this image kind of looks better, and it's basically closer to the true image for phenylalanine and cryo-EM. So to summarize the above, we are faced with the problem that there are not enough tools for validation in cryo-EM. And there was this idea that by using averaging, it's possible to mine 3D shapes from EMDB database and to create a library. And this can be used as a reference to develop tools for validation for map model validation. So with this in mind, we derived a library of map-motifs. This is how it looks like. It contains 155 motifs, one for each rotamer, and they are derived for 7 resolution bands. They are periodically updated as new entries are coming in. And currently they are from 2 angstrom to up to 4 angstrom resolution. And they are accessible at this address on our FTP area. Then based on this library, we developed a tool for local map model validation, which is available. It's called 3D Strudel. It's available from PyPy and GitHub, and also it's integrated in the CCPM software suite. It's not yet released, but I think it will be released probably next week. And then to help users to visualize the results of this program, we also built a plugin for ChimeraX, which is very user-friendly and can be used for analysis of the validations results. So now a bit more about how we've done that. First about the library itself. So I will not go into details here, but the basic idea is here to select high quality entries, then cut them into pieces which correspond to amino-acid residues, classify them. So each piece is, so we have classes within each of them, the conformation it's close, and then superimpose and average them together and obtain the library. Like that's in simple words. So here, like again, how this library look like. This one is for the resolution range between 2.5 and 2.8 angstrom. In total, there are 7, from 2 to 4, and from the address. So now, when we had the library, the idea for validation is like to isolate the map for each residue and to compare it with each canonical motif in the library, and to assign a score which basically reflects how similar is the residue here to each of the motifs in the library. And then you can look at the data and say, is it similar? Does it look like the residue which was modeled here, or maybe it looks like something else, or it doesn't look like any of them too much. So this principle was implemented in the 3D Strudel program. How it works, it takes a cryo-EM map, atomic model, and the Strudel library, does the calculations, and produces a matrix which is of the size number of residues by number of motifs in the library, and also it has associated metadata for the alignments and some other data, and it's saved as a CSV and JSON file. So this program is available again on GitHub and PyPy. It's a pip installable program. Now, having this program, like the data is good, but it's difficult to look into it as a user. So we decided to create a graphical interface, so the user is able to locally look at each residue and inspect whether the model was built correctly or not. So for that, we used the ChimeraX program as a base because it has a good interface for displaying models and manipulating models. So we built a plugin for it. The plugin is available on the ChimeraX toolshed page, and also can be installed directly from ChimeraX. And also, if you install 3D Strudel using the CCP/EM, it's not yet available, but it will be soon. This plugin will be also installed. So this is how it looks like. So this is the ChimeraX window, main window, and this is the Strudel plugin. It has, I'll go quickly through data what is shown here. So here is the map which was validated, the model. Here is displayed the resolution range for the Strudel library. If the validation was done against several libraries, it's a drop-down menu, you can navigate between them. Here is the chain of the of the protein chains. Here is the average score for the chain and the percentage of outliers. Now, here is a scatter plot, which basically represents each dot, it's an amino acid residue. And if the modeled residue, it's close to the motif of the same type amino acid residue in the library, then it's green. And if it's significantly more similar to something else, then the other motif, well, residue type is shown in red. So here was modeled glutamine, and but it says that it's more similar to glutamic acid. So all these dots are clickable. When you click on them, the corresponding part in the atomic model and the map will be shown here. And this plot will display all the motif types from the library. So here it's clicked on this one. It's a histidine, it's shown here. And it scores best with a histidine motif from the library, which is good. And here are the shown other amino acid residue types motifs. And with the scores, which between the map here and the motif. These are also clickable, and if you click on them, like on this one, it will be shown the motif from the library here. So it will be shown green as here. Green is the one which was modeled. If you click on some others, they will be shown in gray, and it will be red if some other motif was scoring better than the modeled one, and it will be shown in red here, only the first one here if it's present. So now how to use it. So here is validation results for one of the early COVID entries, which had several issues, like two main issues on chain A. So one is here, and we could see it straight away with Strudel because there is a drop in the scores here, and also there are two outliers. And if we look at it, can see that the chain was mistraced, and this is why the scores are low. And yeah, basically this residue is outside the map. So you could see that. And here there is another area of the sequence which has a stretch of outliers. And yeah, so keep in mind this one. So we move, this is the same. So if we look at the motifs, at the residues here, so this one, it was modeled as a leucine, and it scores best with tryptophan. So here it's the details, and here is how the model and the map look like. So obviously it doesn't look like a leucine here, so there is quite a lot of additional map. So it makes sense to, well, visually you can see that that the tryptophan could fit actually here, and if turn on the motif, then it fits quite well. So when I looked at the sequence, I could see that there is actually only one tryptophan in this area. I showed only a small part, but it's not, there are no other tryptophans here. And it's residue 116. So we can hypothesize that here it's actually not residue 907, but should be 916. If so, by looking at the sequence, you can see the proline, which is another quite prominent residue. So it should be like after two residues, right? And it's this one. And when I clicked on it, I could see that proline indeed scores the best here. Not so much difference between the one which was modeled and proline, but still it scores better. And you can see that it actually makes sense. And here are some other residues, like this one, like aromatic residues are in general easier to spot. So I look at them. And here, like 911, it's asparagine, but scores best with aromatic residues. And it again makes sense. So using the Strudel program, we could easily spot that here it's a register shift of 9 residues. And later on, it was an update published for this entry. And it was consistent with what we've seen here. And here is validation for the new model. And here are the same positions in the map. And now it's all much better. And there are no more outliers here, except this one, but here is just the form map. So to summarize, we've created a library of canonical motifs for amino-acid residues, implemented a program which uses it for validation, and created a visualization plugin for ChimeraX. And they can be downloaded separately, but also soon everything will be installable using CCPM with the standard distribution. Okay, thank you.